Hello students, hope you all are fine and doing well. So welcome to my channel. Once again, I extend a very warm and cordial welcome to Dr. Sandipan Sengupta's Modern Biology, meant for BSc and MSc degree courses and various competitive examinations like CSIR-NET, GET Life Sciences, NEET examination, IIT-JAM, and so on. So I'm over here with a new lecture session of microbiology, and you are listening once again to your favorite Dr. Sandipan Sengupta, a university assistant professor of microbiology and biotechnology. So in our previous microbiology lecture session, we were discussing about the history and development of microbiology, and we had taken a thorough look over the historical perspectives pertaining to the syllabi of GET Life Sciences Microbiology. So we have well gone in very detail through the major historical perspectives of microbiology as per the syllabi of GET Life Sciences Microbiology. So this is going to be a continuation of the previous session, and in this part of the lecture session, we'll again take a thorough look over the contributions of prominent microbiologists. Because of the contributions made by these microbiologists, which we'll be discussing in very detail today, microbiology arose as an independent discipline. So after listening to today's talk, that is students who are pursuing BSc and MSc degree courses in different disciplines of life science will be able to write up their BSc and MSc degree examinations, and it would also help the aspirants of competitive examinations, particularly GET Life Sciences Microbiology, to answer a lot many questions which are asked from section 1 of the syllabus of GET Life Sciences Microbiology. So this is going to be a very thrilling and very exciting lecture session. And after listening to this entire talk, in which we'll be thoroughly going through the contribution made by each and every microbiologist, if you still have any doubt, please do leave your Gmail ID at the comment box to receive a free PDF or free study material on the topic. But be sure that you are leaving your mail ID in the comment box only after you have listened to this entire talk. So after listening to this talk, if you still have a doubt, please leave your mail ID in the comment box to receive a free sample note on the topic. So let's start, and do not skip any segment. It's going to be very engaging, very informative, and hope you'll enjoy the lecture session. So let's start with an introduction to microbiology. So what do we mean by microbiology? We have already discussed, but still to recap, what is the science all about? So microbiology is, that is the branch of biology that involves the study of microorganisms. And who are these microorganisms? And which organisms would you prefer classifying as microorganisms? Microorganisms are those small organisms which cannot be observed with the naked eye and whose magnified images can be observed under a microscope. So what cellular dimensions do these microorganisms have? So all those organisms which have a cellular dimension of 1 micrometer or less are classified as microorganisms. So organisms having cellular dimensions of 1 micrometer, that is having cellular dimensions in the micrometer range, are classified as microorganisms, while larger organisms are classified as macroorganisms. The group includes, that is the study of a wide range of organisms, the most important one being bacteria, algae, fungi, protozoa, helminths in some worms and nematodes, and viruses. So among all these, that is bacteria, algae, fungi, protozoa, helminths in worms, and viruses, only bacteria have a prokaryotic mode of organization, while all others, other than viruses, except viruses, have a eukaryotic mode of organization. Now see, microbiology is a combination of actually 3 words compiled and together to form microbiology. Micro, which refers to the small size of the organisms included in the group. Bio means life, and logy is the study of these small microscopic or submicroscopic particles having very small dimensions lying in the micrometer range. Now, among that is, as I said, the group includes the study of bacteria, protozoa, algae, fungi, that is, helminths and worms and nematodes and viruses. Bacteria are prokaryotic, the others are eukaryotic, while viruses form an exception. So can you tell me why is it so? Doesn't it sound very interesting? Why are viruses an exception? Viruses are indeed an exception because they have a special mode of life cycle, and they are acellular in nature. They are constructed around a nucleocapsid core, which is often surrounded or not surrounded by an envelope. The nucleocapsid core is often surrounded by an envelope or may not be enveloped. And what about the life cycle? They have a special type of life cycle in which there are two phases: intracellular, when the viruses are present inside their host cell and host organisms, and extracellular. So viruses show metabolism and significant metabolic activity only in their intracellular phase of life cycle, and hence are considered as living objects only when present inside the living hosts. In their extracellular phase, they are not considered as living organisms. Hence, therein lies a separate system for taxonomy and classification of viruses, which is known as ICTV, International Committee for Taxonomy of Viruses. We will have a separate study about each and every group of microorganisms in very detail in different lectures. Now, the history of microbiology, that is, progressed gradually and can be divided into 4 different periods. It began in and around 1600 and kept on progressing. So the history can be divided into the discovery era, that basically deals with the discovery of microorganisms, the transition period, that deals with the theory, with the controversy prevailing or circling around the theory of abiogenesis versus the doctrine of biogenesis, the modern era of microbiology, in which not many developments took place, which we'll go through each and every in very detail while going through the contributions of all those microbiologists who have worked in the golden era of microbiology, sorry, not modern era, that is. And lastly, the modern era of microbiology. So presently we are going through the modern era of microbiology, or it's the era of molecular microbiology. Now let's have an independent look over the major contributions pertaining of different microbiologists pertaining to each and every era of microbiology. Antony van Leeuwenhoek. So when we think of microbes, the first name that comes to our mind definitely would be the name of Antony van Leeuwenhoek, because he was the one who discovered microbial life. So Antony van Leeuwenhoek, his contributions are confined to the discovery era of microbiology, and by in and around 1665, Antony, by 1665, he had constructed his that is, constructed his own microscopes, which could magnify in between 50 to 300 times, and he had several such self-constructed microscopes available. And under one such microscope, he was the first to observe a collection of microscopic particles, which he called as animalcules. And what were these animalcules? When investigations were carried out, these animalcules were found to be a collection of bacteria and protozoa. So microbial life was discovered by Antony van Leeuwenhoek in 1665, while he observed and precisely described animalcules as which were a collection of bacteria and protozoa. And thus, because of his immense contribution, he is considered as the father of bacteriology and protozoology. When we speak of speak of prominent microbiologists, now the next name which comes to our mind must be the name of Louis Pasteur. So Louis Pasteur's work were confined to the golden era of microbiology, and the golden era of microbiology, that is the war spanned across five, that is decades. It started in and around 1860 and lasted till 1910, and spanning across 5 decades, that is almost more than 50 years, and were, that is, dominated by the contributions of two major microbiologists, Louis Pasteur and Robert Koch. Now, first, let's have a look over the that is contributions made by Louis Pasteur. Then we'll switch on to the over to the contributions of Robert Koch. So Louis Pasteur, that is first, that is in 1861, could resolve the controversy that was circling around the theory of spontaneous generation or abiogenesis. So there were two theories about the origin and of life and microbial life. One was the theory of abiogenesis. By abiogenesis, what is abiogenesis? By abiogenesis, we mean to refer to the fact that life may originate or is able to originate from non-living matter or inanimate objects. That is the theory of spontaneous generation, when life arises spontaneously out of inanimate matter objects, or the theory of abiogenesis. But that is on contrary was the theory of biogenesis, according to which microbial life and biological life can arise, or arises rather, only from pre-existing microbial life and pre-existing life following multiplication, reproduction, and cell division. So was the theory of biogenesis, and Louis Pasteur was a huge supporter of the theory of biogenesis and had a firm belief in the theory of biogenesis. So in order to disprove the theory of abiogenesis and put the controversy of spontaneous generation to rest, he designed his own experiments. And for his own experiments, he ingeniously designed his own special flasks, which had their mouth openings as shaped in the form of U-tubes. And these flasks were called Swan-NET flasks. And hence, the experiment carried out by Louis Pasteur to disprove the theory of spontaneous generation using his self-designed Swan-NET flasks are called as Swan-NET flasks. Now, let's have a look at what he did in order to disprove the theory of spontaneous generation. So, these were a series of experiments performed by Louis Pasteur using his own designed Swan-NET flask. So he brought in a Swan nutrient solution in a Swan-NET flask, sterilized it or boiled it for a brief period to remove the associated microbes, and after this, the flask was left open to air for a definite period of time. However, following incubation, after a certain time passed, after a few days, there was no growth of microbes in the flask, or microbes were not found growing over the nutrient media present in the flask, although the flask was left open to air. So what might have been the reason behind this? The microbes which were present in the air or along with the air tried their best to enter and reach the nutrient solution, but were trapped and had settled down in the U-shaped neck of the flask due to gravity. So the microbes settled in this portion of the flask due to gravity and could not access or reach out to the nutrient solution. The nutrient solution was still sterile. However, when the U-shaped neck portion of the flask was broken and the nutrient solution was made accessible to outside microbial life, air-carrying microbes soon could reach out the nutrient solution, and the sterile solution was used by microbes to divide, grow, reproduce, multiply, and to produce new microorganisms, and microbes could now be observed in the flask. The microbes, the clean air carried microbes which accessed the nutrient solution, which reproduced, multiplied, which divided by binary fission, and gave rise to new microbes. So by this, Pasteur concluded that it's only pre-existing life that can give rise to new life via reproduction, multiplication, and cell division, which is so called as the theory or the doctrine of biogenesis. So in 1861, as a result of Pasteur's Swan-NET Flask experiments, the theory of abiogenesis or spontaneous generation of life from non-living objects was finally brought to rest by Louis Pasteur, resolved, and the theory of biogenesis was proved to be and pre-existing life was it had been already experimentally shown by Louis Pasteur that pre-existing life is the source of new life that can give rise to new biological life, following reproduction, multiplication, and cell division. So this was the first introduction of Pasteur. That is, he proved the doctrine of biogenesis and put the theory of spontaneous generation to rest. Now, what was the next contribution that was made by Louis Pasteur in the golden era of microbiology? His works were not only confined to his own Swan-NET Flask experiment, restricted to his Swan-NET Flask experiment, but he kept on studying the process of the conversion of grape juice to wine. So initially, that is during that time, around 1837, there was, in between 1837 to 1860, there was a huge controversy circling around the conversion of grape juice to wine. And this conversion was thought to be a chemical process which was thought to be occurring due to the breakdown of protein albumin. However, Louis Pasteur was the first microbiologist who studied the process in very detail and could point out that no, it's not a chemical but a biochemical process which involves the fermentative role of a microorganism, eukaryotic microbe, that is yeast, biologically named Saccharomyces cerevisiae. Hence, he introduced the concept of fermentation, and he proved and introduced and proved that the conversion of grape juice to wine is a biochemical process involving the work of eukaryotic microbe yeast, biologically named Saccharomyces cerevisiae, and the process being made, or the process is called as fermentation. That is what its name as fermentation. So thus, Louis Pasteur introduced the concept of fermentation. Now, while after this was proved by 1860, Louis Pasteur kept on studying, and he could, that is, observe or find out the spoilage of wine that was caused by microorganisms. So Pasteur could understand the role of microbes in spoiling wine and in causing spoilage of products. So next to fermentation, Pasteur introduced the concept of pasteurization. So what was pasteurization? It was not only wine, but it was also milk and milk-based products which were studied in very detail and whose spoilage was noticed, and Pasteur tried his best to find out the role, that is, rather pointed out the role of microbes in spoiling milk and Pasteur introduced and devised a method for pasteurization. That is, according to pasteurization, milk and milk products can be prevented from this spoilage by mild heating at a temperature much below boiling, that is only at 62.8 degrees Celsius for a brief span of around 30 minutes. So if the milk, if you heat the milk and milk products for a brief span of 30 minutes at a very low temperature of around 62.8 degrees Celsius, this would destroy the population of unwanted microorganisms and would simultaneously not ruin the taste of milk. And this was the concept of traditional pasteurization, which was introduced by Pasteur in late 1860s and between 1860 1870. And pasteurization, that is, became a commercial process which was applied, which started, that is, every milk and milk-based industry, product industry, started applying the process of fermentation for preventing the spoilage of milk and milk products. So nowadays, lot many changes have taken place in the traditional method. Number of, that is, the process has been modernized, and a large number of alternatives, that is, transformed, that is, methods have been introduced. But still, Pasteur, the traditional method introduced by Pasteur, was the first that was to be such method that was introduced by Louis Pasteur, named as pasteurization, to prevent the spoilage of milk and milk products without ruining or spoiling the taste and quality. Now, let's have a look. Pasteur, we'll again get back to Pasteur, because Pasteur also had a main interest in the one interest in also that is managed to make out immense contributions in the field of medical microbiology, because it was only Louis Pasteur who actually pointed out that microbes which could spoil wine, microbes which could cause spoilage of milk, could also make humans fall sick. We'll get back to that, and that is having a close relation with the work of Robert Koch of Germany. Robert Koch and Koch postulates. But before that, we'll take a look over the contributions made by Lord Joseph Lister. So in the modern era of microbiology, in and around 1900 to 1910, that is, Joseph Lister made immense contributions. He was impressed by Pasteur's work, which could show the involvement of microbes in causing disease. And Joseph Lister pointed out that it was only microbes which were responsible for carrying wound infections. So wound infections, indeed, according to Lord Joseph Lister, were also caused by microorganisms. And to prevent the spread of wound infections, or the Lister introduced the concept of sterilization. So he devised a method which involved a fine spray or mist of carbolic acid or phenolic acid that was sprayed in operation theater to sterilize the air and to make it free from microorganisms. Moreover, Joseph Lister also popularized the use of phenol over wound surgical dressings to prevent the wounds from getting infected by microorganisms. So Lister was the first to introduce the concept of antisepsis, or the use of physical and chemical agents to promote sterilization and to make objects free of microbes, disease-causing microorganisms. And because of these contributions, Joseph Lister is considered as the father of antiseptic surgery, who could first introduce the concept of sterilization and could first introduce the use of physical and chemical agents to remove unwanted microorganisms from the surface of inanimate objects and from other materials. Robert Koch, next to Louis Pasteur, another name which comes to our mind while going through the history of the golden era of microbiology, is obviously the name of Robert Koch. So Koch was highly impressed by Pasteur's work, and impressed by the work of Pasteur, Koch was the first to point out the role of microorganism as a pathogen in causing disease, which soon became popular as the germ theory of disease. So according to germ theory of disease, the diseases are caused by microbes which act as pathogens, and this was proved while Robert Koch isolated anthrax bacillus, or the causative agent of anthrax disease, that is Bacillus anthracis, in 1880. In 1882, that is Robert Koch could isolate one more pathogen, which was the causative agent of tuberculosis. So he isolated Mycobacterium tuberculosis. So anthrax was the first pathogen, Bacillus anthracis, which was discovered and which was invented by Robert Koch as the causative agent of diseases. And this laid the foundation of the germ theory of disease. And next to this, Koch also, that is, worked in the field of etiology and laid a series of postulates which are called as Koch postulates. So what do we mean by etiology? Etiology is the real investigation that involves the causal that is the cause behind a specific disease, and also involves establishing a fine-tuned relationship between the causal organism, disease-causing organism, and the that is a disease-causing pathogen and the host. So the etiology, that is, Koch made immense contributions in the field of medical microbiology and devised or introduced Koch postulates to study the etiology behind every pathogenic disease that is still encountered today in the field of medical microbiology. We'll have a look over Koch postulates, but before that, let's have a look over 2 or 3 more contributions of Robert Koch. He also perfected the technique of isolating pure culture of microorganisms. So it was only Robert Koch who could first perfect and who could first introduce the technique of isolating axenic culture, or single population of bacteria from a mixed population. So he perfected the technique of isolating axenic culture of microorganisms from a mixed population of bacteria. And Koch, next to this, popularized the use of gelatin as a solidifying agent. So Koch was the first to introduce the first solidifying agent, that was gelatin. However, gelatin suffered from or had a number of drawbacks. Gelatin was later found by, investigations were made by one of the assistants of Koch, that is Fanny Hesse, who pointed out that gelatin is not a suitable solidifying agent, for it gets degraded when it's attacked by a hydrolytic exoenzyme secreted by cells called, or by bacteria called, gelatinase. Moreover, gelatin gets liquefied at a temperature below that is above 25 degrees Celsius. It quickly melts out when the temperature rises above 25 degrees Celsius. So instead of gelatin, that is the assistant of Koch named Fanny Hesse popularized the use of agar as a solidifying agent, and that again became a major landmark in the field of microbiology. Agar is the most popular solidifying agent that is still used to prepare solid media. So Fanny Hesse pointed out that agar is a mixture of complex polysaccharides made up of galactose, galactopyranose units joined together, that is obtained from different members of red algae like Gelidium, Gracilaria, and agar is hardly attacked and can be degraded by bacteria. So this was the one reason for which agar was preferred by Fanny Hesse, and the second reason which he showed was that agar has a very high melting point of 96 degrees Celsius and sets in at or gets solidified well in between 40 to 45 degrees Celsius. So because of these reasons, agar could replace gelatin, and agar became the most common solidifying agent that is still in use to prepare solid culture plates, media plates. Now, there was one more assistant of Koch named Richard Petrie, who was the first to introduce Petri dishes, the dishes which all microbiologists still use today in laboratory, in our labs, to prepare culture media. Now let's have a look over Koch postulates one by one. So one of the most prominent or major contribution of Koch was the introduction of Koch postulates. There were 4 series of postulates, and the postulates are still in use to study the etiology or to investigate the cause behind a specific disease and to establish a causal relationship between the disease-causing pathogen and the host individual. So what are the 4 postulates of Robert Koch, which he could then postulate and are still in use even today? Let's have a look. Now, Koch said and conducted his experiments and could prove which were experiments were conducted in favor of each and every postulate, and every postulate was proved indeed by Robert Koch. So the first was that the disease-causing pathogen should be present only in diseased individual, but not in healthy. So for this, the specimens, the tissue specimens or fluid specimens were collected from 2 organisms. One was diseased and the other was healthy individual. And when the specimens were studied under microscope, only the diseased specimen could show the presence of disease-causing pathogen. This was the first postulate of Koch. The second postulate was, or Koch next to this postulated, that the pathogen, isolated pathogen, that is, must be grown in pure culture. So that is the second part, and this was indeed moved experimentally by Koch, and he perfected, meanwhile, the technique of growing, that is, or the isolating pure culture of microorganisms from mixed population of bacteria. So every pathogen, according to Koch, must be isolated pathogen which is encountered in the field of medical microbiology, must be grown in pure culture. Now, in the third step, Koch said and even did, that is, now Koch said and performed too, that the pathogen, let the pathogen be introduced into an experimental organism, and let's check whether the pathogen causes the same disease. So when this was performed, that is, and the pathogen, isolated pathogen, was introduced into an experimental organism, the organism was found to contract the same disease. So this was the third postulate of Robert Koch. And lastly, Koch said that the experimental organism, that is, where should be screened for the pathogen must be re-isolated or isolated from the experimental organism, and after isolation, that is, its identity must be identified, and when identified, when its experiments were done to check its identity, it was found to be same as that of the previous or original pathogen. So these are the 4 postulates of Robert Koch, collectively called as Koch postulates, which are still in use to study and to investigate the etiology or the cause behind every disease that is encountered in the field of medical microbiology till date. Edward Jenner is one more name which definitely comes to our mind when we speak of immunization and when we speak of the golden era of microbiology. So, but much before the golden era of microbiology, Edward Jenner, by 1798, that is by the beginning of 1800, had introduced the concept of immunization. So what Edward Jenner did, Edward Jenner could isolate, that is, lesions of cowpox, that is, lesions of cowpox virus from diseased individuals, that is, from group of diseased individuals suffering from cowpox were, that is, they were screened, and the cowpox, that is there, and from the pus of diseased individuals, cowpox lesions were back, that is, virus antigenic preparation was isolated, and the isolated cowpox preparation was introduced into a group of healthy individuals, and it was interestingly found that the healthy individuals that is, the isolated preparation, which was the antigenic preparation from cowpox virus, was protecting the healthy individuals from smallpox, and thus the isolated, that is the preparation from smallpox, from cowpox, sorry, was indeed an antigenic preparation, which when introduced into healthy individuals, was able to stimulate an immune response, could induce the production of proteins called antibodies, and thus made them or turned them immune to smallpox and protected them from the disease. And this was the concept of vaccination or immunization introduced by Edward Jenner. And the word vax contained in the term vaccination. So this was immunization or vaccination, which has been modified and is still in use and has gone through a large number of developments. And there have been that is innumerable developments that have taken place in the field of vaccine biology. But this concept was introduced by Edward Jenner. So by immunization and by vaccinization, by vaccine, we simply need to refer to an antigenic preparation that is known to stimulate an antibody response or an immune response when introduced in the and provides protection against disease. Now, next to Edward Jenner, let's have a look at the contributions of Alexander Fleming. So contributions of Fleming were, that is, not confined to the, that is, golden era of microbiology, but by the end of golden era, Fleming, that is, had started his studies over gram-positive bacteria, Staphylococcus aureus, and the Alexander Fleming, could come up with his first advent in the form of first antibiotic, penicillin, in 1928, which almost marked the end of the golden era of microbiology and marked the beginning of the era of molecular or modern microbiology. So Alexander Fleming that is studying over the colonies of gram-positive bacteria growing in the because the colonies, these were the colonies of Staph aureus, and somehow accidentally the plate was that is by chance contaminated by a fungus named Penicillium notatum. However, interestingly, Fleming observed that the fungus was secreting a substance, the mold Penicillium notatum was secreting a substance that was inhibiting the growth of gram-positive colonies. And when screened and characterized, the substance was found to be penicillin. And this was the first antibiotic introduced by Fleming in 1920. Penicillin was produced by the mold Penicillium notatum, and this was the first antibiotic introduced by Fleming. And this marked the beginning of the era of antibiotics. And next to Fleming, there were a series of antibiotics like streptomycin, tetracycline, which were discovered from a large number of low G plus C actinomyces gram-positive members, that is streptomyces. Now, next there was the next name which comes in the list is the name of Paul Ehrlich. So Paul Ehrlich could introduce the concept of chemotherapy. And Paul Ehrlich's work in the field of chemotherapy, the golden era of microbiology ended and the era of modern microbiology or molecular microbiology began. So by early 1900s, Paul Ehrlich had started investigating the action of dyes that is used for staining which are used for staining bacterial, that is microorganisms. So the action of such dyes that is thoroughly investigated by Paul Ehrlich against common pathogens. And interestingly, he found a dye called trypan-red active against the trypanosome pathogen causing African sleeping sickness. So trypan-red dye was found by Paul Ehrlich to be detrimental and to be active against, was found potent enough in destroying the African pathogen causing African sleeping sickness, and because of its property, it was named as magic bullet. And Paul Ehrlich's work continued, and in 1910, Paul Ehrlich discovered arsenobenzole or benzol derivative, that is salvarsan, that is, or arsenobenzole, a chemotherapeutic agent that marked the beginning of the era of chemotherapy. And arsenobenzole, discovered by Paul Ehrlich, was discovered by him as a treatment against a spirochete that causes syphilis and is named as Treponema pallidum. So the advent of chemotherapeutic agent arsenobenzole or salvarsan by Paul Ehrlich marked the beginning of the era of chemotherapy, and chemotherapy soon became very popular. And what do we mean by chemotherapy? The use of specific chemicals as chemotherapeutic agents to kill pathogenic microorganisms and pathogenic organisms without causing harm to the host individual is the science called as chemotherapy, which was introduced by Paul Ehrlich with the advent of salvarsan or arsenobenzole to treat syphilis caused by spirochete Treponema pallidum. So with this marked the beginning of the era of chemotherapy. Fleming's work marked the beginning of the era of antibiotics. Jenner's work was could indeed introduce the concept of vaccination and immunization. Now, these were the major microbiologists who actually contributed during the discovery era, the transition period, the golden era, and helped microbiology to microbiology in pushing it ahead and in its emergence as an independent discipline. Now, by that is after the end of modern microbiology, there began an era called the era of golden era of after the end of golden era of microbiology, there began an era called the era of modern microbiology or molecular microbiology, in which virus was discovered, in which a lot of developments took place. Amplification, the method to amplify a gene was discovered. The method of DNA sequencing was discovered. That is, some of the major developments which took place in the beginning of the modern or era of modern microbiology or molecular microbiology was the discovery of first virus particle by Ivanovsky. He was the first to crystallize and observe a plant virus particle named TMP, tobacco mosaic virus. And this was the first virus particle to be crystallized, whose structure was elucidated, and indeed the first virus to be discovered. Now, Vinogradsky is considered as the father of soil microbiology. Beijerinck developed a technique called enrichment culture technique, which is still in use and which then marked the beginning of a method called enrichment, that is selective isolation and screening of a particular microorganism from a group of microorganisms. So selectively permitting the growth of a particular microorganism is that that is terminology or the concept of enrichment that was introduced by Beijerinck. Now, other developments, these were followed by still more developments like the advent of PCR, polymerase chain reaction, that is used to amplify or make several thousand copies of a single gene under laboratory conditions. That was discovered by Carey Mullis. That is in the era of molecular microbiology in 1985, although it was years later. And now soon, that is the method of DNA sequencing also became popular, while Gilbert and Sanger introduced methods to sequence the genetic material present in the atom, that is to work out the sequence of DNA. So these were the contributions of almost all major microbiologists who contributed in the discovery era, in the transition period, in the golden era of microbiology, and whose contributions pushed microbiology ahead and paved the way for emergence of microbiology as an independent discipline. Hope you have enjoyed the lecture session. For more such videos, do subscribe my channel. I'll cover each and every topic meant for that is pertaining to the syllabi of that is GATE Life Sciences, syllabi of CSIR-NET, and my videos are mostly based on the topics of microbiology, biochemistry, molecular biology, and biotechnology. That is modern biology. So hope you have enjoyed the lecture session. So see you all with that is in the next lecture session with a new topic of microbiology. Till then, a very goodbye to you all.